Hello guys, today's video we're going to tackle about photosynthesis. What is photosynthesis? First, from the Greek word photo means light and synthesis means to put together. In short, light to put together. Photosynthesis. So, photosynthesis. Have you ever thought about the food that you take from the farm to your plate? The process of photosynthesis, which transforming sunlight into energy that sustains life. Let us explore the intricate details of this important process. From sunlight capture to nutrients synthesis. Here, take a look of this carbon dioxide cycle. This is the cycle of our carbon dioxide. This is here. Here is our trees, and here physicochemical. Physicochemical involve the principle of basic and chemistry. Refers to the fact photosynthesis involves both physical and chemical. Physical aspect is capturing light energy, and the chemical energy aspect is that conversion of that energy into chemical bonds. So, photosynthesis of organic compounds. Using the periodic table of contents in chemistry, we are going to use the elements of. Because of combination of chemistry, we're going to use the elements, which this one. And what is anabolic process? Anabolic process is those that build complex molecules from simpler ones and so requiring energy. For example, carbon dioxide and water. So, with that combination, we're going to have C6H2. We're going to have this one converted into glucose. That is what we call complex organic molecule. So we have the chemical equation for photosynthesis here. And so if we are going to ask what is enzyme? Enzymes, proteins that catalyze speed up chemical reaction.
Or Theodore Wilhelm Engelmann, usa ka ika-19 nga siglo nga German nga nakahatag dakong kontribusyon sa genbayo, ilabing sa pagtuon sa photosynthesis og microorganisms. Nailhan si TW tungod sa iyang pagpa-experiment gamit ang mga microorganisms og kahayag sa pagtuo sa photosynthesis. Sa usa ka experiment, migamit siya og spectrum sa kahayag aron mahibal-an kung unsang mga wavelength labing epektibo sa pagdagan sa mga proseso. Sa photosynthetic sa mga tanom sa tubig, kini pagka-discover nga ang pipila ka mga wavelength ilabing ang pula ug asul nga kahayag gikan sa kinihanglan sa photosynthesis. Cornelius van Niel, 1897 to 1985, on the basis of studies with purple and green sulfur bacteria, showed that photosynthesis is a light-dependent reaction in which hydrogen from undissoluble compound reduces to a from sulfur. Si Cornelius van Niel, usa ka American microbiologist nga nakahatag og dakong contribution pod sa genbayo. Pagtuon sa photosynthesis og microbial metabolism, si Van Niel labing nailhan tungod sa iyang ground locking nga nagibuhat sa biochemical nga process sa photosynthesis sa bacteria. Gisugyot niya ang teorya sa photosynthesis nga naglambigit sa pagbahin sa mga molecule sa tubig imbis sa carbon dioxide nga usa ka nagpatibabaw nga pagtuo ni adtong panahona. Joseph Priestley in 1770 on the basis of experiments showed the essential role of air in growth and green plants. A mouse kept in closed space could get suffocated and die. But if a main plant is kept on bell jar, neither candle will distinguish nor will the mouse die. He concluded that full air produced by animals is converted into pure at by plant. Si Joseph Priestley, usa ka ika-18 nga siglo sa Ingles na siyentipiko nga nailhan sa iyang mga contribution sa chemistry, education. Sa biology, siya sa pagka-discover ni adtong 1774 nga iyang gitawag nga depocalistated air. Ang mga na-experiment ni Priestley sa tanom mitultol sa mga panabot sa photosynthesis nga nagpakita nga ang mga tanom napagawas sa oxygen. Julius von Sachs in 1854 shows that green part in plants produces glucose, which is stored as starch. Starch is the first visible product of photosynthesis. Si Julius von Sachs, usa ka ika-19 nga siglo, kontribusyon nga German nga nakahatag og dagkong kontribusyon sa Gen Bio. Labi na sa pagtuon niya sa mga tanom, nailhan siya sa iyang panukduking bahin sa nutrisyon sa tanom. Labi na sa iyang mga investigation sa papel sa tubig ug mineral. Sa pagtubo sa tanom, naghimo usab siya og importante nga mga contribution sa atong pagsabot sa photosynthesis, metabolism sa tanom ug ang impluwensya sa mga hinungdan sa kinaihan sa paglambo sa
Where does photosynthesis occurs? So before answering that question, let's have a brief discussion of some parts of the plants that will lead to where does photosynthesis occurs. So, just like humans, plants also breathe. So if we humans have our nose as our inhalanan, then plants also have. And that's what we call stomata. So stomata is a tiny opening where they breathe in the carbon dioxide. But photosynthesis doesn't happen there because sulod ana is na yung mga like walls nga galinya, and that's what we call mesophyll. And inside the mesophyll, naadira si chloroplast where photosynthesis occurs. So chloroplasts are green plastids which functions as the site of photosynthesis in eukaryotic photoautotrophs. So kani si eukaryotic photoautotrophs kay mo na siya ang mga organisms nga nag-use og sunlight to manufacture their own food. And that process is ginatawag natong photosynthesis. Mga example ana is kato si plant or algae. So within the chloroplast, there is a membranous system consisting of grana, the stroma lamellae, and the fluid stroma. So kana si membranous system or membrane system kay muna siya ang in charge sa pag-capture and pag-store sa light energy. So photosynthesis has two stages or process. The first one is light reaction and the second one is dark reaction. So si light reaction is muna siya diri nga process nga mag-trap sa light energy aron makaproduce og ATP and NADPH. So kani si ATP and NADPH kay muna siya ang energy storage and electron carrier or donor molecule. While sa second stage nga si dark reaction kay mupod ni siya ang mo-utilize sa ATP and NADPH aron mo reduce sa carbon dioxide. And during that process, si carbon dioxide kay mo reduce dayon into sugar or glucose. So now, let's move on to the pigments involved in photosynthesis. So before that, si pigment is mo kini ang mo-absorb sa light o mo po kini ang mohatag og color sa plant. So we have here the photosynthetic pigments. So naa diri si mga pigments such as chlorophyll A as blue-green, chlorophyll B as yellow-green, carotenoids as yellow-orange, and xanthophylls as yellow. So diri sa first picture, makita nato ang measure sa pag-absorb sa mga pigments sa light. So sa second picture is mopud na ang rate of photosynthesis. Og i-combine ni nato si first and second picture, makita nato sa third picture kung asa nga pigment ang mas kusog mo absorb sa light, and that is the chlorophyll A. So therefore, chlorophyll is the primary pigment of photosynthesis that absorb light. Then si accessory pigment pod is mo ni siya ang mga mo help sa chlorophyll molecule, and also mo avoid nga mahitabo ni si photooxidation such as chlorophyll B, carotenoids, and xanthophylls. So kani si photooxidation kay mo ni siya ang cause sa high intensity sa light or UV rays na directly igo sa tanom, which can damage or destroy the plant. That's why si accessory pigment is ang in charge nga mo prevent ani.
Hi Ma'am, hi classmate. I am reporters of Group 2. My topic is all about the light reactions. The light reaction, also known as light-dependent reaction, are a series of chemical reactions that occurs in photosynthetic organism. This reaction takes place in a chloroplast that require light energy to convert water and carbon dioxide into ATP and NADPH, which are energy-rich molecules used in the next stage of photosynthesis. So, during the light reaction, the two photosynthesis system, which is the photosystem 1 and the photosystem 2, are play a crucial role. So the photosystem 2 is absorb light energy and uses it to split water molecules, releasing oxygen byproducts. The electrons from photosystem 2 are passed through an electron transport change, which generates a proton gradient across the thylakoid membranes. So this proton gradient is used by ATP synthase to produce ATP. So meanwhile, the photosystem 1 is absorb light energy and uses it to energize electrons, which agent of north, which are transferred to NADP plus to form NADPH. So the NADPH is an important reducing agent used in a Calvin cycle to convert carbon dioxide into glucose. And overall, the light reaction harnesses light energy to generate ATP and NADPH, which provide the energy and reducing power needed for the synthesis of carbohydrates during the Calvin cycle.
So, unsa ng electron transport system? So, the electron transport system is the process to transfer the electron between molecules, generating energy used to produce ATP. In short, ang electron transport system kay process siya to produce ATP. And ang photosystem is a complex of proteins, pigments, and other molecules found in the thylakoid membrane of chloroplast in plants and algae. In short, ang photosystem kay complex siya sa proteins, pigments, and other molecules makita nimo sa chloroplast in plants and algae. So, na ito type sa photosystem. Ang photosystem 1 and photosystem 2. Ang photosystem 1 kay ma-absorb siya light wavelengths longer than 680 nanometer while ang photosystem 2 kay absorb siya shorter wavelengths than 680 nanometer. So, ito taba sa process sa light emitting reaction of photosynthesis. So, ano yan? Thylakoid. Ang chloroplast po siya na electron molecules na photosystem 2 electron transport chain and then photosystem 1 C again, and the last is ATP, or we call adenosine triphosphate. So we start ang step 1 kay photoexcitation. Ang photoexcitation is striking molecule of chlorophyll inside of photosystem 2. Chlorophyll is the pigment actually absorb the sunlight. So ang chlorophyll kay siya ang absorb og sun energy gikan sa sunlight. When money yang giab energy sunlight giabsorb sa chlorophyll, and when chlorophyll absorb the sunlight, that energy is transferred to an electron within photosystem 2, and it proves that electron gain energy and it become excite. So when ang chlorophyll kay mo absorb siya energy gikan sa sunlight, ang kaning energy mo transfer siya into an electron gikan sa photosystem 2, and it causes that electron gain energy and it become excite. So mo ni ang electron kung gain ng energy kay mo excite, so mo gitawag na siya photoexcitation. And ang step 2 kay makita na siya nato siya sa electron transport chain. When that electron in photosystem 2 becomes energized, it can no longer stay in photosystem 2. So if ang electron kuno kay ma-energize, di na siya mo stay sa photosystem 2. It gets passed down a series of other membrane proteins called the electron transport chain. So when ang electron kay ma-excite, so di na siya mo stay sa photosystem 2 mo. Bali na ba siya sa laing membrane protein tawag na to electron transport chain. So mo ni. And then as that electron jumps down the ETC, it releases time energy into the thylakoid. So if ang electron kuno kay muadto sa ETC, ang ETC kay mo release siya some energy padung sa thylakoid, and it also going to help to pump some hydrogen ions into the thylakoid space. So muna hydrogen ion na gi-release sa thylakoid, and nato sa step 3 itawag na to of photolysis, the process of splitting water. To survive photolysis, it takes that water splits it into hydrogen, oxygen, and some hydrogen electron ay oxygen electron yeah. So na ni H2 plus H2 plus O2 plus electron. So ang ganing oxygen kay mo obvious naman na mo ato ni sa air na mo natong kanon ramboy ta na mo na sa byproduct. So ang electron, ang kanang electron kay mo ni siya ang kaning nabilin sa photosystem 2 kay mo ni yan i-transfer ang kaning yan mo transfer siya sa photosystem 2, and then ang kaning hydrogen kay continue to build up inside of the thylakoid space. So mo continue gyapon siya build up. Ang galing mo continue gyapon siya build up sa thylakoid space kaning mga hydrogen ion along with the hydrogen ion. So ano to sa step 4. Ang step 4 kay gitawag na to chemiomeiosis. Ang ganing mga hydrogen kay mo at mo naog siya through sa ATP synthase or adenosine triphosphate synthase. So chemolysis process of pushing the hydrogen ions down the gradient through ATP. So, muna. And then, ang step 5 kay tawag na to reduction. Reduction. The hydrogen going to use to facilitate a reduction reaction. So there is a molecule called ADP. ADP. Called ADP. Those hydrogen ions are going to help convert the ATP to a really potent molecule that we call ATP. So, ang kanindang DP, kato daw mga hydrogen ions are mutabang siya to ang mo convert sa ADP to para mahimo siya ATP, hydrogen ion, ADP, and ATP. Yan ang step 6 na to ay ni kani nanapod siya sa ikaduhang electron transport chain. Process of the electron used to facilitate another reaction similar happening over here. NADP plus going to reduce we call NADPH. It is going to be used to fuel the next set of reaction called light independent reaction. So, ang ganing electrons kay naog na wagi na siya sa electron transport chain na naog siya. May mo siya NADP mo reduction i-check nga to may mo siya NADPH. So, muna na ang step 7 kay itaog na to reduction. So, magbalik similar siya ay tabo sa step 5 nga ganing reduction. So,
good morning, everyone. I am going to discuss the fascinating work of photophosphorylation, and we'll explore the differences between cyclic and non-cyclic photophosphorylation. What is photophosphorylation? Photophosphorylation is the process of generating ATP using light energy during photosynthesis. There are two main types, cyclic and non-cyclic. The differences between cyclic and non-cyclic photophosphorylation. Cyclic photophosphorylation. Number 1. It is formed by photosystem 1 independently. It is because cyclic photophosphorylation is driven solely by photosystem 1. Number 2. An external source of electrons is not required. It is because it does not require an external source of electrons. Number 3. It synthesizes only ATP. It is because the primary product of cyclic photophosphorylation is ATP. Number 4. It occurs only in stromal or intergranal thylakoids. It is because it occurs in the stromal or intergranal thylakoids of the chloroplast. Non-cyclic photophosphorylation. Number 1. It is performed by collaboration of both PS1 and PS2. It is because the non-cyclic photophosphorylation involves both photosystem 1 and photosystem 2 working together. Number 2. The process requires an external electrons donor. It is because this process relies on an external source of electrons, typically water. Number 3. It synthesizes ATP and NADH both. It is because the addition to ATP non-cyclic photophosphorylation also generates NADPH, which is a reducing agent used in the Calvin cycle of carbon fixation. Number 4. It occurs in the granule thylakoids only. It is because it occurs specifically in the granule thylakoids of the chloroplast. Let's talk about cyclic photophosphorylation. In this process, electrons from photosystem 1 are excited by light energy and pass through the electron transport system. However, instead of being transferred to NADPH plus to form an NADPH, these electrons are cycled back to photosystem 1, resulting in the production of ATP without generation of NADPH or oxygen. Maong ginatawag siya nga cyclic photosphorylation kay mubalik naman siya sa form niya. Cycle na siya balik. Non-cyclic photosphorylation. In non-cyclic photosphorylation, electrons are excited by the light energy and pass through the electrons transfer system chain, ultimately leading to the synthesis of ATP and NADPH. Importantly, water is split during this process, releasing oxygen as a byproduct. So, maong ginatawag po niya nga non-cyclic photosphorylation, kaya from the word non-cyclic, dili siyang balay siyang form.
Good evening, everyone. So let us tackle about the chemiosmotic hypothesis. So what is chemiosmotic hypothesis? This hypothesis is a process that energy is released through a series of oxidation reduction events when electrons are transported through an electron transport system. This energy can be used by specific chain carriers to the move of hydrogen ions or H plus or proton. So, what ang H plus kay mo siya yung nasa sulod, which is nasa diri sa gawas yung sulod. Then, next is the one who proposed this hypothesis is Peter D. Mitchell. So, Peter D. Mitchell in 1961. Again, Peter D. Mitchell in 1961. So, ATP synthesis is linked to development of proton gradient across the membrane or thylakoid and mitochondria. So, mo ni siya si ATP synthesis. So, chemiosmotic hypothesis. The chemiosmotic hypothesis process compiles the movement of ions through an electrochemical gradient. A semi-permeable membrane facilitates this movement. The process of chemiosmotic hypothesis requires a membrane, ATPase enzyme, gradient of proton, or pump of proton. The chemiosmotic hypothesis explains that the complete process of synthesis ATP in the chloroplast. ATP synthesis is connected on the inflammation of proton gradient in the lumen. So there are three main events found during the process of chemiosmotic hypothesis. One is photolysis of water towards the thylakoid membrane. Number two is transfer of H plus to stroma to lumen as electrons move through EPS. NADP reductase, the reaction occurs toward the stroma side of the membrane. So, mao to ang tulo ka main events are found during the process of chemiosmotic hypothesis. So next is transfer of H plus from stroma to lumen as electron moves through the photosystem. So ang kuan ani niya is kani. So mo ani siya. So ako lang siya butang dira sa picture, which is ang first ana kay makita natin sa picture dira no dira. So ang first ana kay ang katong mga H plus kay dire naa sa stroma, which is mo na ang stroma na sa gawas nya. Ang kaning circle nga pink is thylakoid membrane, mo na gitawag nga thylakoid membrane nya. Ang kaning nasa sulod mo ni ang katong sa sulod, which is mo ni ang kaning mga H plus, which is ang pangalan niya is lumen. So si stroma di ay kay mas low ang iyang kuan, ang iyang hydrogen ions, which is ang H plus, mas low siya compared sa lumen nga nasa sulod nga ang hydrogen ions niya is mas higher than the stroma. So the process of this chemiosmotic hypothesis is then ang kaning FO, which is ang kaning nasa babaw, FO kani makita ra nato dira sa picture ako lay ipakita. So FO is mao ni siya ang kanang present on the inner side of the lumen. So na siya sulod sa lumen, like sa sulod siya sa circle, which is kani sa sulod namurag na siya diri ba. It is embedded in the membrane and forms a channel that carries out the diffusion of protons across the membrane. So, ara siya una mo agi ang H+. Then mo paingon dayon siya sa F1, which is the present on the outer side. So gawas siya sa stroma na siya nga side sa gawas, which is kaning stroma sa gawas. Nya mo ni ang thylakoid membrane. Okay? So makita ra na to dira ang picture dira nga makita ra na to og asa dapit si stroma nya asa pod si thylakoid membrane og si lumen. So lumen makita ra to sa sulod na dayon. Kaya ako sa explain na the present of the outer side of the stroma and converts the ADP. ADP means adenosine diphosphate and ATP is adenosine triphosphate. This protrudes on the outer surface of the thylakoid membrane. So outer side siya, mom ni thylakoid membrane kaning circle nga pink, which is naa diri si F1 sa gawas ni thylakoid membrane. So na siya dira nya ang FO kay naa sa tunga ug kanang sa sulod sa lumen. Sa sulod niya ba? The next so the ATP generated and immediately use of biosynthetic reaction. The synthesis of sugar and fixation of carbon dioxide or CO2 takes place in the stroma. So first is photosystem 2, which is kani. Lagyan po na to sa chart dira. Photosystem 2 nga it presents the membrane. When sunlight, like ang energy sa sunlight falls in the photosystem 2, the antenna molecule absorbing the sunlight yields energy. So inika-absorb nila, so ma-form na siya nga energy. Nya dayon mo transport the system nya mo to ang H+ accumulates the lumen, so mo papunta din mo ato rin siya sa lumen. Then electron goes to the photosystem 2 to the primary electron acceptor. Mo na siya ang first nga kuan dayon. Then the plastoquinone or hydrogen carrier and electron carrier. Plastoquinone takes hydrogen from the stroma and releases into the lumen. So kana si plastoquinone kay iyang kuaon ang mga H+, which is si hydrogen ions gikan ni stroma pag naday i-release dayon siya ni lumen. So si stroma mo to ang outside, which is akong giingon ganina nga si stroma nasa gawas na dayon si kuan. Then si lumen is nasa sulod, which is si stroma has a less hydrogen. Hydrogen means is H+. So mas katong para sa akong giingon ganina nga mas low ang hydrogen ions ni stroma kaysa ni lumen. Yeah, it also transfers electrons to cytochrome B and F. So mo transfer dayon siya ni cytochrome B and F. So nasa tunga. So chart again. Dayon releases sufficient energy in the lumen, generates proton motive force. Electron release energy comes to the ground state and moves to photosystem 1 via plastocyanine. So, ang electron release energy, so ang electron mo release of energy daw comes to the ground, the state of move, then mupunta dayon siya sa photosystem 1, which is mo na yung photosystem 2, no? And then, cytochrome B and F, niya dayon. Mo na ni ang photosystem 1, PS1 and PS2. Kung in short. Na dayon. Next is the photosystem 1 again, mo-absorb siya light, nga dayon, mo-create na po siya energy, then the electron moves to NADP and via FT to NADP. So mo to ni-absorb siya light para sa photosystem 2, hindi mo punta din siya sa NADP, which is na diri. So again, chart again. Kita ka pa ito sa chart, tara ako siya ibutang, siya edit. Dayon, kita na nato kadiri mo siya maklaro. So inika ko man sa photosystem 1, yung mo absorb og light, yung mo create og energy, then mo paano siya sa NADP. It clearly shows the process of NADP reduction of NADPH at photosystem 1 in the case of non-cyclic phosphorylation. Then, electropotential gradient is generated due the high H plus in the lumen. So, si electropotential gradient kay nag-generate daw siya due sa high of hydrogen ions sa lumen. It is important to break the gradient to release the energy. So, importante daw nga ma-break ang gradient to release an energy. Ako rin yung translate. Then, the concentration gradient breakdowns occurs. Then, this is due to the proton movement. Proton movement occurs from lumen to stroma via ATP synthesis. So, ang proton daw, which is si H+, move siya from gikan sa lumen to stroma na dayon muato din siya sa ATP synthesis. Lumen, gawas dere, stroma, and then ATP synthesis. Dayon, ATP synthesis has two portions. One faces to stroma, outer membrane or head portion side, while the other faces of lumen side is the integral part of FO. H plus comes outside with the help of FO. When it comes outside, the works as a motor and changes the structure of F1. ADP or adenosine diphosphate with organic phosphate makes the adenosine triphosphate. This consequences completes the process of the chemiosmotic hypothesis. So, si NADP reductase enzyme is located on the stroma side of the membrane. Protons are necessary to reduce NADP plus to NADPH plus H plus, and these protons are moved from the stroma. Within the chloroplast, protons in the stroma decrease in number, while protons accumulate in the lumen. It causes decrease of lumen pH and creates a proton gradient across the thylakoid membrane. So again, the meaning of NADPH is so in conclusion, this process of chemiosmotic hypothesis concludes that the whole ATP formation with reference to photolysis of water and NADP reduction and electron transport system, it involves a different location. Different electron and protons carriers and accomplish the activity. ATP forms as the product of the whole procedure, utilize the biosynthesis of sugar and provides energy to the proper functioning of the plants. So that's all. Thank you, everyone.
Hello everyone, so today we are going to discuss about the Calvin cycle. So Calvin's cycle can be described into three stages or parts, which is carboxylation, reduction, and regeneration of RUBP. So first thing that happens is that carbon dioxide enters the cycle, which is this one. So see, carbon dioxide came mosulus the cycle. Carbon dioxide is going to react with ribulose, so this one. So carbon dioxide came will react sa ribulose, which is this one. Catalyzed by the enzyme riboscosamon, hina siya diri, and thus going to turn into 3-phosphosylserate represented by the symbol of PGA. So this one, symbol of PGA. So that is the carbon fixation. So RUBP is a molecule with 5 carbon atoms. On the carbon 1, we have phosphate group, which is this one. We have a phosphate group. And on carbon 5, we have another phosphate group, so that's RUBP. Can you see it? Which is RUBP. RUBP is a molecule with 5 carbon atoms, which is this one here. On carbon 1, we have phosphate group, and on carbon 5, we have another phosphate group, so that's RUBP. Now there are 3 of them, and they are going to react with 3 CO2 molecules. Now, when one molecule of CO2 reacts with one molecule of RUBP, we're going to get initially a 6. So 6, this one, 6 carbon molecule, but they're going to be broken into 2. So 6 divided by 2 is equals 3. So we have 3 carbon molecules. PGA is a 3 carbon molecule, and it's going to have 1 phosphate group. So the 1 phosphate group is this one. And it's going to have 1 phosphate group, so that if we keep track of a total number of carbon, if each RUBP has 5 carbons, 3 of them has 15 carbons. And then we just have a 15 carbons here. And then when we're going to add 3 carbons from the 3 CO2 molecules. So 15 carbons plus 3 CO2, which is this one, equals 18. So we have a total of 18 carbons. So thus there are 6 3-phosphosylserate molecule. When you see enzyme kinase, that enzyme kinase is used to transfer a phosphate group to a molecule, and so this the next steps requires ATP. We're going to use 6 ATP molecules. So we put 6 ATP because we need 6 ATP molecules. To phosphorylate 3-phosphosylserate, and now we have 1 3-phospholyserate. So we still have 3 carbon molecule, but now we have a phosphate group on a carbon 1 and a carbon 3. As you can see here, the PGA kinase is an enzyme that catalyzes the conversion of 3-phosphosylate into 1 to 3 by phosphosylate. So ATP gives up a phosphate in order to become an ADP. So ADP to give up a phosphate in order to become a ADP. And that phosphate is transferred to this molecule. This you can see here, we already have 2 phosphate groups. So anytime you see a kinase enzyme, it catalyzes the transfer of a phosphate group from one molecule to another. Now in the next steps, we are going to use NADPH. So as you can see here, we put NADPH. So we're going to use this one to reduce this one. into G3P. 3 phosphate, so NDPH is going to convert, so NDPH is going to convert to NEDP. Plus, we put plus here. Now, we need 6 molecules of NADPH to do this reaction. So, nibutang tag 6 NADPH and 6 NADP will be catalyzed by the G3P dehydrogenase enzyme. So, this enzyme, as it suggests the words dehydrogenase, and it removes hydrogen from NADPH. Now at this point, we're going to get 6G3P molecule. One of the 6G3P molecules is going to be used to produce sugars like glucose and fructose. And things like that, the other 5 G3P molecules is used to regenerate the 3-RUBP biphosphate molecules. So always keep in mind the total number of carbon here is 15. Each G3P molecule has 3. Each G3P molecule has 3 carbon atoms. This one. And it has a total of 15 carbons, which is what we started with. So the net result, the Calvin cycle converts 3 molecules of CO2 into 1 molecule of 1G3P. And that's the result of Calvin cycle.
Good day, so we're going to tackle about the C4 pathway or the Hatch-Slack pathway. So this pathway was worked out by Hatch and Slack of 1965 to 1967. This is mainly operational in plants growing in dry tropical region like maize, sugarcane, sorghum, etc. And this pathway for a stable product is a 4-carbon compound, oxaloacetic acid or AAO, so called as pathway. Plants have Kranz anatomy or vascular bundles are surrounded by bundle sheath cells arranged in wreath-like manner, characterized by a large snow of chloroplast, thick wall impervious to gases, and absence of intercellular spaces. The primary acceptor is a 3-carbon molecule phosphenol pyruvate present in mosophyll cells, and enzyme involved is PEP carboxylase. Next is OAA formed in mesophyll cell forms 4-carbon compound like malic acid or aspartic acid, which is transported to bundle sheath cells. And bundle sheath cell, it is broken into and a 3-carbon molecule. The 3-carbon molecule is returned back to mesophyll cells to form PEP. The molecule released in bundle sheath cells enters the Calvin cycle, where enzyme Rubisco is present that forms sugar. And now this is the diagram of the C4 pathway. Thank you.
Photorespiration is a metabolic process that occurs in the plant during photosynthesis. It is the light-dependent process where oxygen is added to ribulose 15 biphosphate instead of carbon dioxide, leading to the release of carbon dioxide when concentration of oxygen in the environment is increased from typical 2 to 3% to 20%. Photorespiration becomes more prevalent. This process completes with the normal photosynthetic pathway and decrease the overall rate of photosynthesis. In photorespiration, the enzyme Ribisco, which responsible are responsible for fixing carbon during photosynthesis. Beans with oxygen instead. This results in the formation of two carbon compound and the release of carbon dioxide. And unlike normal photosynthesis, photorespiration does not produce energy-rich molecules of contribute the ano, contribute the production of sugar.
This pathway involves chloroplasts, peroxisomes, and mitochondria. Photorespiration do not occur in C4 plants. Are the differences between in situ plants and C4 plants? In situ plants, the leaves don't have crans and autonomy. In C4 plants, the leaves show crans and autonomy in leaves. In situ plants, rubus photorespiration occurs. In C4 plants, photorespiration does not occur. In situ plants, rubisco is the first acceptor of CO2. In C4 plants, PP is the first stable product. In situ plants, PGA is the first stable product. In situ plants are adapted to all climates. In C4 plants are adapted to tropical climate. In situ plants, mesophyll cells perform complete photosynthesis, while in C4 plants, mesophyll cells perform only initial fixation. Pathways involves chloroplasts, peroxisomes, and mitochondria. Chloroplasts are organelles found in plant cells that are responsible for photosynthesis. During photosynthesis, chloroplasts capture light energy and convert it into chemical energy in the form of ATP and NADPH. These energy-rich molecules are used to drive the Calvin cycle, where carbon dioxide is fixed into organic compounds like glucose. Peroxisomes are small organelles involved in various metabolic processes including lipid metabolism and the breakdown of toxic substances. In plants, peroxisomes play a crucial role in photosynthesis process that occurs in C3 plants but not in C4 plants. Mitochondria are organelles responsible for cellular respiration, where organic molecules as glucose are oxidized to product ATP. In plants, mitochondria also play a role in the breakdown of organic acids produced during photosynthesis and other metabolic processes. So, the pathway involves chloroplasts, peroxisomes, and mitochondria is essential for photosynthesis, cellular respiration, and various metabolic processes in plants. While photorespiration occurs in C3 plants due to the characteristic of rubisco and environmental conditions. C4 plants have evolved mechanisms to minimize photorespiration and improve carbon fixation efficiency.
I will be discussing about the factors affecting photosynthesis. So there are four things that affects the photosynthesis, which is light intensity, carbon dioxide, temperature, and water. So in the light intensity, when the light intensity increases, taas po ang rate of photosynthesis until maabot siya sa saturation point. So ang saturation point is if mo taas even na mo taas ang light intensity, dili na mo taas ang rate of photosynthesis because of limiting. And then for the carbon dioxide, if mo taas ang rate sa carbon dioxide,
Tapos mo-increase po ang rate sa photosynthesis until maabot siya sa compensation point. So ang compensation point kay the point which reach in a plant when the rate of photosynthesis is equal to the rate of photosynthesis. The point reached in a plant when the rate of photosynthesis is equal to the rate of respiration. And then for the temperature, it does not influence directly ang rate of photosynthesis, but at the higher temperature, enzymes activity is inhibited due to dehydration. Dehydration of enzymes, which affects the direct reaction. So, direct reaction is the light-dependent process in which the sugar molecules are formed from the carbon dioxide and the water molecule. So, ito ma-influence the rate of photosynthesis, but maka-affect siya sa photosynthesis. So, if too cold ang photosynthesis, ang productivity can slow down siya. As if cold po siya, if hot po siya, di siya mo produce. Dili siya ka photosynthesis sa water pod. So another water. So if ang tubig sobraan nimo butang og tubig ang plant, the rate of photosynthesis is not proportionally increased. So, 1 siya. So, kay after manggod sa saturation point, kay dili na ka mag-need of water. So, di siya makatarong. But, importante po ang water kay if dili mo ang rate of photosynthesis, maybe lack of nutrients ang plant. And ang need nga nutrients is water and other more. Okay, if kanang naglakag nutrients nga water, kay magka-cause of damage sa chlorophyll. And about sa kaning ang gingon about kanina nga limiting or maong dili mo increase ini ka about sa saturation point, kay about ni sa kaning giingon ni Blackman's law. So ang Blackman's law kay states that if a chemical process is affected by more than one fraction, then its rate will be determined by the fraction which is nearest to its minimal value. So meaning when a process depends on a number of factors, its rate is limited by the phase of the slowest one, minimal slowest. So if tagan sila, mag-depend sila sa kinhinayan, pinakahinay. That's all.